Can you imagine spending years in jail for a crime you didn't commit? Unfortunately, that has been a reality for many people. If DNA fingerprinting had been used in the crime scene investigations, would it have changed the verdict for those who were wrongly convicted? DNA fingerprinting or profiling is yet another application of biotechnology, and it has become extremely valuable in crime solving.
Forensic science is the link between scientific evidence and the courtroom.
DNA evidence played a major role in the O.J. Simpson murder trial, as well as in rape and other criminal investigations. But what is DNA evidence or DNA fingerprinting, and how is it obtained? The cells in a person's body contain chromosomes. Each of these chromosomes contains many sections of DNA with a unique pattern. With the exception of twins, no two people have the same DNA pattern. DNA fingerprinting is simply the technique of identifying that unique DNA pattern of a person. It provides a profile and positive identification of the person in a way that is similar to an actual physical fingerprint.
PCR stands for polymerase chain reaction, and it's a marvelous piece of technology which has literally revolutionized all of biotechnology. It is what is currently being used for DNA fingerprinting, forensic diagnostics, pathogen diagnostics, and medicine and field application data. The principles of PCR is based on DNA replication, and essentially what this technology has the ability to do is amplify or clone fragments of DNA. That is what PCR is capable of doing. Okay, so in the event that you're watching CSI or any of these shows and you hear about PCR, you know what it stands for. Polymerase chain reaction.
If the DNA profile from a crime suspect matches the profile from a sample of blood, hair, semen, for example, found at the crime scene, there is a very high probability that they came from the same person, the suspect.
We usually work on hairs, fibers, but we mainly do biological fluids. The fluids that we work on are blood, semen, vaginal secretions, and saliva. We work on saliva, but not much because we don't do a lot of tests that we used to do on saliva. So basically the saliva is used like, if there was oral sex in the case, then we will look at the saliva to see if there's any sperm in saliva.
Though it does not provide 100% certainty, DNA fingerprinting is more reliable than eyewitness accounts or circumstantial evidence. Here in Trinidad, there are dozens of unsolved criminal cases, such as the murder of Akil Chambers. Perhaps, if DNA fingerprinting had been used at the crime scene or on evidence obtained at the autopsy, authorities would be further along in their investigations and closer to identifying and arresting a suspect. DNA fingerprinting is also a very valuable technique in other types of investigations. It is used to identify people killed in natural disasters, plane crashes, or tragedies like the September 11th attacks. It has helped solve paternity cases and to confirm the identity of kidnapped children, thereby allowing for their return to their parents. Many cases do not involve DNA evidence, and often there is not enough of it to convict a suspect. However, if accurate and untainted DNA evidence exists, many juries find it important in making their decisions. It is highly possible that if the search for and analysis of DNA evidence had been done at our local crime scenes, the books might now be closed on many of these unsolved cases. Indeed, given the number of unsolved crimes in our country and the wider Caribbean, perhaps the time has come for DNA profiling to be used as the main tool in criminal investigations in the region.